Thank you. Just share my screen here. Can you see my screen?
Yes, yes.
Okay, great. Well, thank you. Thank you for this great meeting this morning, and happy birthday, Terry. So is this a dog or a muffin? Now, this sounds like a trivial question to us, but actually this is a fairly complex problem. And there is a very interesting paradox around this problem known as Moravec's paradox. And it goes like this, that things that we humans can, automatically do without thinking, that seems very easy to us, and we don't need even to be aware of the computation, like perception, are actually much more difficult for machines. And things like playing chess or Go that seem very difficult to us, we now have AI systems that can solve them very well. So despite the fact that we have deep learning that can solve these very complex problems, we still do not have a good system that, if it has never seen these dogs and these muffins at this scale, it can solve them. It cannot generalize very well. And this is what's called as Moravec's paradox, and it has many interesting stuff, not just in perception, but in robotics and so on. And the question is why? Now, neuroscientists have been trying to understand visual perception, which is essentially a process where we have to infer the latent causes in the world. Like in this case, we have to, when we look at an image and it's noisy photons hitting our retina, we have to infer that that image is caused by a muffin that we can go and eat versus a little dog that we can pet. And this is a very complex problem, and it mathematically, and this is what neuroscientists have been trying to solve. And one of the major, if not the main approaches of physiologists for decades has been trying to understand representations. How is information about visual images represented in different stages of visual processing? And there's many heroes and successes in this story, and this goes back to one of the most famous people is Hardline, who he did this phenomenal work in the 40s where he would use spots of light to study retinal ganglion cells and little line segments, as you can see here. And later on, Hubel and Weasel did these incredible experiments where they discovered orientation selectivity, columns, and so on. But a lot of these discoveries, as you will know, I'm not going to play the movie, but you can find it on YouTube, where David Hubel describes his discoveries were by serendipity. It wasn't, as you can see here, they were using spots of light. They were like Terry, students of Kufler or postdocs with Kufler. Kufler was a retinal physiologist, so they were using these spots of light, trying to study the visual cortex. And one day, by mistake, one of the slides that they were using that had these spots of light, it was creating an edge between the background and the slide. And then they figured out that this neuron was very well driven. And then, they were smart enough to figure it out that it wasn't the spot of light, but it was this edge that was driving the neuron. Then they discovered orientation selectivity. There are many cases like that. If you look at the discovery of grid cells or place fields, these were not discoveries that were driven by either a theoretical normative model or by some sort of very, maybe there was some intuition in all of them, but, serendipity in science plays a very prominent role, and in particular in the visual system. And the reason why it's so difficult to have big discoveries like that, and they happen rarely, is that if you take, for example, a monitor that is 100 by 100 pixels, and you just create black and white images on it, you can create in the order of 2 to the 10,000 patterns. And this is an intrinsically very dimensional input, and there is essentially more, almost an infinite number of patterns that you can generate on the monitor to try and find, let's say, the optimal stimulus that would drive a neuron very well, or trying to understand a multidimensional tuning function to try and get some interpretability or understanding of how information is represented, and sort of match the successes of what we have in the retina, or even in some areas like the visual cortex, or other areas higher up like face cells and so on that were discovered by Charlie Gross in frontotemporal cortex. So here today I will present to you a method that we developed called Inception loops, and which I think is a promising direction to try and solve this problem in a systematic way. And I think the method that we've developed is general enough. It can be used for any system that one wants to try, where you can enter the era of large data, where you have the capability of collecting very large scale data. And the idea is to build an in silico avatar of, in our case, the visual cortex. And I will go into the details of how we do that and show you some data. So Inception loops have 4 major stages. The first stage is that you have to do large scale recordings, or you have to, it could be applied to any other system, but you have to have a system, a platform that you can collect very large scale data. In our case, in these particular experiments, we collect in the order of 5 to 10,000 neurons, and we'll show 5,000 static natural images from the internet, in this case, ImageNet. And each image is presented for 500 milliseconds, and we interleave this with blank images of 300 to 500 milliseconds. This data we'll talk about today were collected with 2-photon imaging. We have experiments like that, with silicon probes in monkeys, and so you can apply this to any system. So in these particular experiments, we did dense imaging for reasons that will become apparent in a minute, but the idea here is that you record from all these neurons in a little chunk of the visual cortex. In this case, was layer 2-3 of primary visual cortex of the mouse. The second step is you have to build a predictive model. And essentially what you want to learn how you can predict the responses to any one of these 5,000 neurons well, given an arbitrarily new, never seen natural image before. So we want to learn if you want a predictive model, or in a more technical term, we're using system identification methods to build this predictive model that learns the nonlinear mapping from pixels, when pixels is a natural image, to responses. And in this case, the response, you can think of it as a vector that is a 5,000 neurons or 10,000 neurons. Now, as Terry said, we're in the era of large data, but also deep learning. And what deep learning excels at, for reasons that are still people trying to figure out, is that if you have large scale data, in this case, we have 5,000 images and we have 10,000 neurons, we have in the order of millions of training points that can train these networks. Like in this case, it's like 50 million training points. So the idea here is that you build this model that matches the response of the brain. Now, this is not an implementation level model. It's a system identification model that you're just reparameterizing the nonlinear transformation using a neural network whose component parts may not bear, they don't have to have any faithful similarity to how the brain does it, but the outcome has to be very similar. And that's what we're optimizing. This is where the loss function is. So you're trying to learn the responses. Now, the beauty of this is that once you have this model of the brain, which we can call an in silico avatar of modeling these 5 or 10,000 neurons, it is no longer just a black box, as Terry was saying. You can open the black box and you can do experiments in silico that are essentially infeasible to do in the real brain without having this model. And I will describe you one of these experiments. There are many experiments one can do, and we're working in different experiments, but this is one in silico experiment that it's almost impossible to do in the brain. And just before I describe that, this model has some, it's a deep convolutional neural network, but because this is a mouse and you cannot train the mouse to fixate, but also has behavioral components. You cannot behavioral to the model. And in this case, the way the model is decomposed, it has this core, there's a deep convolutional core where all the 10,000 neurons are reading out from the same core, and you have a linear readout for of each of these 10,000 neurons. But you have an embedding of the nonlinear transformation from pixels to the feature space is common. And the idea here is that these are all neurons in layer 2-3 of primary visual cortex. They're doing similar computation, not exactly the same, but similar. So you can learn this function in these two stages. You can learn the nonlinear part through this common core and the linear part for each one of these neurons. And these type of models that we and others have been building in the last couple of years are state of the art in predicting responses to natural images, unlike other previous models that were in some ways more interpretable. As I said before, this model also can take into account the eye movements of the system. Or you can have behavioral if you train the animal to do specific behavior. Here, the animal is running, sometimes it's still. So you can put all this modulation in the network. So you have an end-to-end trained network that can predict the responses of a neuron given a specific behavior. And this is state of the art, as I said. You can read this in the paper, but it can achieve on average in the order of 80% of the explainable variance. But for the neurons that I will talk about today, although the description that I will provide is not just for these neurons, the results apply to all neurons that you have good performance or statistically significant performance, but we will focus on this, a subset of these neurons for the data that I will show you. So as I said, this is not a black box. You can perform this in silico experiments that drive experimentally testable predictions that would have been essentially impossible to do in the brain. And one of them is to ask the following question. You can take one of these neurons that you have a good predictive model and ask the question, what's the optimal stimulus for that neuron? Because you've trained this through a neural network, you can use back propagation, but in this case, instead of changing the synaptic weights like is typically done in machine learning, you can optimize the pixel space to find the optimal stimulus. And I will show you a visualization of this. You start with an arbitrary noisy image, and then you pass it through the network for this target neuron. This is the in silico avatar, and you get some response. Now you can compute the gradient. You can pass the gradient through the network all the way to the pixels and say, how should I change the pixels to increase the response of that neuron? So this is the forward pass, and in the backward pass the gradient, and now you do a gradient step, an ascent if you want. So you go from pixels now to increase the response. And you can do this back and forth many times. You can run this on a GPU and overnight, and you can optimize all these 5,000 neurons, and you get what we call the most exciting image, or the MEI, or the most exciting input. And when you do this, you sort of see something that would make Hubel and Weasel like say, yeah, this is what we've discovered. This is an edge detector. It looks like some sort of Gabor patch. But when we did this for many neurons in mouse visual cortex, and I have to say here that in monkey, when we do it, we get much more of a representation that looks like Gabor and Hubel and Weasel, and possibly would be trained the cat. But when we did this in the mouse visual cortex, we got a lot of these most exciting inputs that were not looking like Gabor filters, more interesting structures like the one I show you here. And when my daughter saw this, my 10-year-old daughter saw this, it reminded her of a Chihuahua dog. Now, of course, I don't think these are Chihuahua dogs, but the point here is that they did not look like they had simple, a lot of them, some did look like Gabor, but there was a decent amount of them that also did not look like Gabor. And I will show you an array of them just to give you the diversity that one gets. As I said, these are ones that were selected to have very good predictive models, but even if you look at the rest of the thousands of neurons, we have decent predictive models, we see the same level of diversity. So here is ones that do look like Gabors or edge detectors. Here are some that look more like checkerboards. And then here are ones that look like sharp corners. This is a black corner on a white background. And here is a corner for a different angle. Here is something that looks like a right angle corner, a white corner on a black background. And this is more like a curved stroke. So we saw this diversity of dictionary of features that our model was predicting that this would be optimal for these neurons. Now, of course, you know, this is just in the model. It could all be an artifact of the model, and there's many reasons to be worried that this is an artifact of the model. For one thing that you have to do as a sanity check, despite, you know, you have to make sure that this is not a solution. And again, as Terry said, you know, surprisingly enough and unintuitively, you know, you can do the following experiment. You can start with a completely different starting image in this high dimensional pixel space, in this case, 2 random white noise patterns and 2 natural images. And then you can run your optimization, and what you see is incredibly consistent across neurons. So it doesn't matter what starting image you start, you find the same solution. Okay, so this gives you some confidence that, you know, if this was not the case, it would be very hard to be convinced to even do an experiment in the first place, to go and check these stimuli. So here we can go now and do the experiment. We can take these neurons. We can find these neurons across days. They are stable across days. This is one thing. But we can go and show now these 150 images back to these neurons. And when we do this, we get these confusion metrics. What I'm plotting here is the real data now from the brain, where we go, because we do chronic imaging, we go back, we find these neurons, we show them all these 150 MEIs, and each row here is the responses to the MEIs ordered according to the prediction for each neuron. So for example, if you take neuron 35, we ordered the MEI, so MEI 35 was the one that was optimized for these neurons. These neurons have, because of the density of the imaging we do, these neurons have essentially overlapping receptive fields. And the fact that you get this strong diagonal and you don't see a lot of responses in the background tells you 2 things. First, that these predictions, you know, we're predicting the right, you know, most exciting image for each one of these neurons. And we also did there's a lot of controls in the paper that you can read, that these stimuli were driving the responses more than a lot of different control. And I'll just show you one of them, stimuli including natural images and other, you know, optimized Gabors and so on and so forth. This again shows how good the model is. Now we are looking at the predicted responses versus the observed responses of these MEIs, and this is one neuron, and you see that you get a correlation of 0.89, and this is the distribution of the correlation coefficients. So these models are very good at predicting the responses to these most exciting images when we show them back. Now, when you look at that, we can also map the linear receptive field of the neurons, and there's a lot of similarity. In fact, mouse, unlike monkey, there's a very much stronger, especially in layer 2-3, linear component of these responses compared to the classical complex cells. So they look more like simple cells, if you want, but they don't look like Gabor. But there is quite a difference also. So here's the most exciting images. Here's the receptive field. The most exciting images have higher spatial frequencies. And when we compare the two, as you can see, the receptive field gives you a much lower response. Now, to the next step is when you look at them, as I said, they don't look like Gabor, and we don't have a good normative model, and we still don't understand what does this representation mean. And this is a problem that is also faced by the deep learning community. They can train a neural network to do incredible computations, but it's not clear what is the normative understanding of this representation. We have this very beautiful normative model by Alsazen and Field that is thought to explain primary visual cortex for animals like cats or primates. And when we did the same type of experience in primates, indeed, we see that most of these neurons do look like Gabor filters, Gabor-like filters for the monkey, but they don't look in the mouse. So to start understanding, yes, how many?
10 minutes.
Okay, thanks. So to start understanding that, we did the following experiment. We took a new set of 5,000 images. I mean, 5,000 was just a number. You can take 5 million images. You'll get to the same conclusion. And now you can crop them at the mask of the most exciting input or the receptive field of the neuron, if you want. Okay, so now we can take these 5,000 images and novel images the model has never seen before and pass them through the model and ask the following question: which image passed is the most exciting, will drive the neuron the most, okay? And when we do that, we get something that looks like this. So this is out of these 5,000 images for this neuron in the model, this image patch was driving it the most. And now, if we look at, sorry, this is the MEI and of the neuron. This is the synthesized most exciting image. And this is in the middle is the best natural image patch. And as you said, you see perceptually at the pixel level, they are not necessarily similar, but perceptually they look very similar. And if you look at it, it happened to be a patch that was part of this image here, that was this minivan. And if I go through a lot of these images, on the left column is the most exciting image, and on the middle column is the most exciting natural image patch, and you see the similarity. When you look at these things, you know, it's not clear how to understand them, but if you now look into image space, you see that you find images at the fairly frequent level, and I will show you the calculation, that drive these neurons not as high, but the high, you know, almost as high as these most exciting images. So here is another one that looks like this sort of, again, tripartite shape, and you see that part of this image looks like this. This is the one I showed you earlier. Here is something that is like an acute angle. Here is something that looks like a checkerboard. Here is one that has this dot with this crescent around it. Here is something that is more elongated. Here is something that looks like a step function. Here's like something that looks like a T-junction. And here is again, another pattern, and so on and so forth. And this is how essentially all these neurons, they are MEIs you find in natural image patches when you look in the order of a few thousand patches that look like their most exciting images. And here's the quantification of this. The most exciting image is always going to be essentially the highest activity that we've ever could find. But then you see the most exciting patch next to it. And if you look at this across many, many neurons, thousands of neurons, about 1 in 2,000 images produces activations that are more than 75% of the MEI, okay? So this tells you the level of sparseness. So if you can think of this like as a sparse level of code, you know, and you have these images being flashed around to the animal, you know. Now, here I haven't talked about surround inhibition effects, and this is a different talk, but this is about this crop, the stimuli that will drive the receptive fields. Now, another thing that when you look at this, you see that they're not random, these things. They seem to be organized into groups. And we have a project with my ex-student Alex Ecker and my collaborator Matthias Bethke and their students. Now Alex has his own lab, where we use a rotation equivariant network to classify these into different neurons. And I will show you some of these clusters. What you see here is a cluster of functional properties of these neurons, and you see that they have the same most exciting inputs are up to a rotation. So they're rotation versions of each other. Here is another one that looks more kind of exotic, has this like eye-like structure, and again, with this black thing around it, and they're like rotated versions of each other. So to summarize, you know, so far, we record from the brain, we build a model, we optimize the model, and then we present them back to the brain, and this is the Inception loop experiment. And I think it has a lot of applications in many different domains in neuroscience and beyond. Now, to really though understand how the brain works, you know, what I've described so far is a systems level identification method that is agnostic to the implementation level details, like the beautiful type of interneurons or the type of level of analysis that Terry was talking earlier. And to you know, emphasize the neatness that we need to go at that level, I will use this toaster analogy. If you have to understand a toaster, there's two, you know, dramatically different approaches to this. One, you could do a structural understanding where you identify all the parts, you try and put them together, you create a toy model. But if it lacks function and computation and behavior, then that model is not going to really lead to a full understanding of how the toaster works. On the other hand, you can build a toaster like, you know, like an iron board here, which I used to do as an undergraduate, where you can make toast. But this toaster has the same, you know, it makes toast like the original toaster, but it's built from different components, and it has a different architecture if you want. But if the toaster, the original toaster breaks down we do not understand how the original toaster works, it will be very hard to fix it. So we need to bring structure and function together. And I will go fast now. We've done some previous work with Xiaolong Jiang when he was in my lab to study the cortical microcircuit architecture by figuring out like many different morphological types of interneurons and how they're related together. And we came up with some rules of how Martinodi cells and other cells like basket cells connect to each other. And we found that if you create, let's say, an architecture that looks even at this level of complexity, which is orders of magnitude less complex than what Nuno and Clay are going to talk about in a bit, you can break it down to a few simple rules. And in this case, we found 3 rules that recapitulate this basic cortical architecture. And the rules are, that the Martinodi cell acts as this master regulator, and this is somatostatin cell, where it connects to every other neuron in vicinity, but 2 Martinodi cells never connect to each other. And the opposite is true for basket cells and PD cells, where they tend to connect within their group or with their family, but they avoid connecting to each other. So when you take these 2 rules and together, you can build a model of the connectivity that fairly well recapitulates the empirically measured connectivity. But in order to bring structure and function together, we started this wonderful collaboration with Clay Reed, Nota Costa, and Sebastian and Jake Remier used to be in my lab. And as part of this Microns project, where in this millimeter cube, we want to bring both structure and function. We imaged the mice at Baylor. We imaged a bunch of these mice, I think around 7 of them, and we imaged about 100,000 neurons per mouse using 2, and in some of them also 3-photon imaging. And we call this mouse that also went through the EM pipeline that Clay is going to talk about, the platinum mouse, which has about, as I said, in the order of 100,000 neurons that we imaged. And we imaged it across 4 visual areas, so we could also study recurrent connections. Here is how the data look like or actually this is even part of the data in this mouse. This is rendering during the presentation of a movie. And now we have all these functional properties, including the MEIs from this model. And as I said, before the travel ban, the mouse could go to Washington, DC, where it was imaged. And we are in the process, and Nuno and Clay will show you the tracing efforts and to try and understand the relationship between structure and function. And I would like to acknowledge my collaborators, Jake Remier, who did a lot of, the work with me and with some students on the functional connectomics experiments at Baylor, Fabian Sin and Edgar Walker, who did all the predictive coding work, the most part of the talk that I showed you, Alex Ecker, Zach Pitko, Matthias Betke, and of course, Nuno, Clay, and Sebastian, who worked with us on the connectomics, and the students and postdocs that worked on the work, again, in particular, Ed Walker and Fabian Sin, who did the predictive modeling, and Talia Muhammad, who did most of those experiments, also Eric Kobos, who did all of the machine learning work in that project. Thank you very much.
Great, thank you very much, Andreas. So yeah, we have a lot of exciting questions here. Unfortunately, we are low on time, so maybe let's do one question, and if you could try to answer the rest by typing, that would be wonderful.
Sure.
So here's a question from Gautam. How well could, say, an avatar trained on simple stimuli, sports, gratings, predict the responses to natural images and movies?
Yeah, so we did that experiment. We have a NURBS paper on that. It's bad. The conclusion is if you train an avatar on natural movies, it responds very well, the responses to gradings and parametric stimuli, at least the ones we've tried. But the other way around, it doesn't work.
Okay, great, thanks. Well, let's take one other quick question. So here, Bin Huan is asking, do you expect more complex MEIs in higher visual layers? maybe.
Yeah, we and there is, you know, so in monkeys we've done these experiments and we do get much more complex. If you go up the visual hierarchy, the ventral stream, you get much increased complexity in the MEIs. In the mouse, we're still working on it, so I don't know the answer to the mouse yet. But you know, it's interesting that in the retina of the mouse, it has more complex features than the monkey. And also now in V1, it seems to have more complex features. So maybe the hierarchy has a different goal of organization than the classical view of increasing complexity as we think in the ventral stream.
Great, thank you. Yeah, very fascinating. Okay, thanks a lot, Andreas.
Thank you. Thank you, everybody.